Let's start by going over the warm-up. So share screen. Let's see. So again, these concepts are going to be in play today as we go deeper into kind of cardiovascular physiology, control of blood pressure and all that. So you really want to feel comfortable with these basic concepts, things that increase resistance to flow. Most people got decreased vessel diameter. Again, that's going to be one of the main ways that your body has to control blood pressure is if you want to raise blood pressure, you can constrict a vessel, which increases resistance, which will increase blood pressure. Increase blood velocity as well. And again, we talked about overall length of blood vessels. For instance, your systemic circuit has like 5 times as much resistance as the pulmonary circuit, just because there's so many more, hundreds of thousands of miles of blood vessels in it. So those are the 3 main things. Systole, AV node was the thing that starts off the systole of the ventricles. We're going to look at that in a little more detail in just a few moments. Yeah, definitely not before atrial systole, because if the atria have to first dump into the ventricles. This one, most people did not get. So this is something that we're going to cover more today as well. The flow in each circuit has to be equal, right? If the flow is higher on one side or the other side, you're going to get congestive heart failure because...
I'm sorry. I swore you had said because it goes farther in the body, it goes faster. Like, because it has a farther journey to go through.
Right, so 2 things. One is flow is not about speed. We talked about that explicitly, that those are 2 different concepts. And second, we talked very explicitly about the cardiac output, the flow on the pulmonary side and the systemic side have to be balanced. The pressure is very different because of the resistance differences, but the flow has to be balanced or else you have congestive heart failure. So unfortunately, when I set up my little iPhone, somehow I ended up getting it so it was not recording audio. So there is no audio for our lecture on Tuesday, which was, I might be able to find like a copy of a video I did from like last semester that covers the basic material and make a link to that. But I definitely did not say anything about one like systemic being faster. So we're going to be looking at this more today, and the idea of cardiac output, which is the flow out of either ventricle, is going to be a key player in our discussion of blood pressure today. Right, and you can just say the cardiac output is the flow out of either ventricle. It doesn't matter if it's the right or left ventricle, they have to be equal. The flow has to be the same on both sides of the heart, or else you're going to get backup pressure on one or the other circuits of circulation, since they each feed into each other. Are there any questions? This is actually an important concept. Are there any questions about this one still?
I think just saying like going out of the heart clarifies, because I got confused just by thinking of like the blood, not that was leaving the heart, like that I understood was the same, but once I started thinking about it like throughout in the actual system, like more moving in the lungs and more throughout the body, that's where I got like a little confused. Okay. So thinking of it of just like what's leaving the heart, that at least clarifies it for me.
Okay. But it is that leaving the heart is going into, it's what's feeding the circuit or draining the circuit. This one, everybody got simple squamous epithelium. Again, in the vessels, you call it endothelium. In the heart, you can call it endocardium, but it's the same thing. This, if you got this one wrong, you should definitely review. Arteries, by definition, leave the heart, right? So if you're leaving the heart on the systemic side, you're carrying oxygenated blood. But if you're leaving the heart on the pulmonary side, the pulmonary arteries are actually carrying deoxygenated blood out to the lungs. So, the veins coming back from the lungs are bright red because they've just picked up oxygen. So veins return to the heart, arteries leave the heart, but whether they're carrying oxygenated or deoxygenated blood depends a lot on whether you're looking at the pulmonary or systemic circuits. So is that clear? All right. So let's stop sharing. And we're going to continue dealing with the heart and dealing with all sorts of other stuff. Question? Okay. I think it's probably worth giving a little, we are gonna come back to the heart and talk a little more about the different parts of the cardiac cycle. But it's gonna be useful to kind of introduce blood pressure in a little more detail right now. So let's do that. And so and again, the aorta is pushing the blood out through the semilunar valve, out into the body. And again, for right now, let's just talk about the systemic circuit. Because when we talk about blood pressure, 120/80 or whatever, we're usually talking about the systemic blood pressure. So obviously the heart is not like a normal pump that runs your dishwasher or something, because it pulses, it pushes, and then it relaxes. Systole, diastole, systole, diastole. So it's pulsing. So when we think about blood pressure, let's let this be like the blood pressure. Again, what are the units we use for blood pressure?
Millimeters of mercury.
Millimeters of mercury. So there's going to be, the systole. Let me make it there. And heart is pushing, and then we got relaxing, and then it's heart is beating, and then it's relaxing. Then it's beating, and then it's relaxing. So obviously when the heart is the ventricles are squeezing in systole, that's when you're really going to be creating this pressure in the vessels. Pressure is just force per unit area. The ventricles are squeezing during systole, pushing blood out. The systolic blood pressure, it can be different. Let's just say it's about 120. And then let me see. So when the ventricle is squeezing, it makes sense that the blood pressure is highest. When the ventricle relaxes, is the heart generating any pressure at all? during diastole? Is there any pressure being generated by the actual heart muscle to drive the blood?
By the heart muscle, I would guess no.
No,
During
no.
diastole.
During diastole, the heart muscle is relaxed. It's when the ventricles are totally relaxed and refilling. So during diastole, the heart is not generating any blood pressure. So your blood pressure starts falling, but it doesn't fall to zero. And we'll talk in a few moments why that is. Typically it falls down, let's say around, somewhere around like 80 or something. So the diastolic blood pressure is lower. And then the next systole, the heart squeezes and it comes back up. But then the next diastole, it starts dropping again. The next systole, it comes back up. And the next diastole, it starts dropping. So it's going up and down and up and down. The pressure during systole is the systolic blood pressure. The pressure that's lower that it drops to during diastole is the diastolic pressure. That's why you always use two numbers to specify blood pressure. Like this would be like 120 over 80. I should use units because it's always diastolic. Oops, it's passing on you. It's always systolic over diastolic to specify the pressure. The reason why the pressure does not drop to zero when the heart isn't actually squeezing is something else is continuing to generate pressure. And what that is going to be the large vessels leaving the heart. Like for instance, the aorta. The aorta is it's basically like a big rubber hose the size of a garden hose. Both the aorta and the pulmonary arteries are what we call elastic arteries. So they're called elastic arteries because their walls are full of elastic tissue. They're really stretchy. So during systole, when the ventricles are ventricular systole, when the ventricles are squeezing, in addition to pushing blood out to the body, they are also stretching out these large arteries, kind of ballooning them out. So yeah, think about a big rubber hose with a lot of pressure inside, and now the diameter is increasing, it's stretching out. So during systole, I should say ventricular systole. So they expand. I don't know, and then when the heart is relaxing in diastole, you're going to have the semilunar valve slam shut, so the blood can't go back into the body. So this valve is going to be closed, and this big elastic artery that's been stretched out is now going to start compressing, and that's going to continue keeping the pressure up and driving the blood forward. So, during diastole, I should say ventricular diastole, during ventricular diastole, that large elastic artery that expanded during the ventricles squeezing are now going to compress. And that compression of these large elastic arteries is going to keep the blood pressure from dropping to zero. That's why your diastolic pressure is not zero. If there were no big elastic arteries, your blood pressure would just go up and boom, down to the floor, and then boom, up and then down to the floor. Instead, it is relatively evened out by these large elastic arteries that, I don't know if, like when I was a kid, we had these things called water weenies. You know, they're basically like surgical tubing that you fill up under a lot of pressure from the hose, and they expand, and then they look like this big salami, and then you can let the water out of it and soak your friends. I don't know if people still play with those kinds of things. But does that kind of make sense? If you took like a big rubber tube and you filled it until it's like a big sausage filled with water under pressure, and then you let go of the end, the water splooshes out. I see some eyes kind of going up like, huh? Does that make sense for everybody? So this is going to be important in a few moments when we also look at the different stages of the cardiac cycle, is realizing even when the heart is relaxed and refilling, there is still blood pressure out in the circuit there that is regardless of the heart. A few other terms we should mention while we're here. This difference between systolic and diastolic, we call the pulse pressure. When you put your finger on a artery and you hear it, you feel it kind of going up and down and up and down. That's that's the pulse pressure. That's the difference between diastolic to systolic, to diastolic to systolic. So that pulse pressure is actually what's generating that pressure difference or diameter changes you feel when you're actually putting your fingers on an artery. That's why it's called the pulse pressure. When we're discussing blood pressure, it's often nice to just have one number instead of two numbers to describe the blood pressure. So we have what's called the mean arterial pressure, which is kind of like the average blood pressure. Again, you're going between systolic and diastolic. Let's say this was 120 and this was 80. Because the blood pressure spends more time lower down your diastolic than systolic, you can't just average them. It's kind of a weighted average more towards the diastolic. So the mean arterial pressure is going to be diastolic plus 1/3 of the pulse pressure. So it's basically the diastolic plus 1/3 of that distance between the systolic and diastolic. And again, this was. Right, so if somebody had a blood pressure of 120/80, what would be their mean arterial pressure map? Plug it in. Yeah, so I got Aaron got there. So diastolic is going to be 80 plus 1/3 of 120 minus 80. 120 minus 80 is 40. So then we could say, all right, 1/3 of 40. All right, let's make this easier. This is 30 plus 10. So this is going to be 10 plus 3 1/3. So 13 1/3. So 80 plus, so it'd actually be like more 93.3 millimeters of mercury. Right. This is used a lot, like especially in studies. or, where they're trying to look at effects of things on blood pressure, they often talk about effect on mean arterial pressure because it just gives you a single number to define blood pressure. As we keep talking about controls of kind of your overall blood pressure, we'll be talking about things that control the mean arterial pressure when we're looking more systemically. I should mention, so I had this equation, pressure equals flow times resistance. This equation works no matter what you're looking at, but we're going to look at it in different contexts. Sometimes we're going to be looking kind of more globally at your entire system. So more globally, the pressure will be like the mean arterial pressure, meaning the overall blood pressure in your body. The flow is going to be the cardiac output. Right, the amount of blood liters per minute coming out of your heart into the circuits, times, the overall resistance of the major arteries leaving the heart. So this would be kind of more global. We're going to spend a lot of time with this basic equation, this basic kind of formulation, looking at how cardiac output and resistance of major arteries, sometimes they call this peripheral resistance. Sometimes we're going to look more local. So that's going to be another thing we're going to do is we'll look at just flow, like local auto regulation, like what's just happening here in your finger or something, in which case we might be looking at flow and resistance and pressure, but not in the big global sense of your whole body. So we'll look at that later, but most of our initial discussions of pressure, flow, and resistance are going to be like kind of the mean arterial pressure, the main blood pressure in your whole system, or systemic side, I should say, cardiac output, the flow coming out of your heart, and peripheral resistance, the kind of resistance to flow of these main arteries leaving. Okay. So now let's go back to the heart. So we were talking about that intrinsic conduction system of the heart and the different steps of kind of the heartbeat. We're going to look at that in a little more detail. Right, and I should mention the heart is pretty amazing. We're not going to get into tons of detail, like anatomically and developmentally, it's pretty amazing. Like your heart starts beating within weeks after you start developing as a little embryo, right? Once you become a ball of cells that's bigger than a certain number of cells, you need to have some kind of circulation or else you can't get the oxygen and nutrients to the cells. So the heart starts beating really quickly, really soon. Long before it looks anything like this, just when it looks like kind of like a little tube that squeezes. And so the heart actually has to kind of develop into a mature heart and keep beating while it kind of actually turns into a mature heart. So it's actually pretty cool watching how that works. The heart also, people have kind of done these calculations, like about 3 billion beats per lifetime. It's kind of crazy if you think about some buy a toaster that can make 3 billion pieces of toast before it finally tuckers out. That's kind of amazing that our hearts last as long as they do. So 3 billion beats per lifetime. And it can't, obviously it has to keep going. At the moment it stops, you're in trouble. Some of the books talk about the heart stripped of its romantic cloak, right? Because most, in most kind of literature and poetry, it's like the heart is where you feel your emotions and stuff. Like in our class, it's going to be a chunk of meat that pushes your blood through your body. So, it's affected by your emotions, obviously. You get excited and you get some sympathetic activation. It's going to speed up your heartbeat or something. But for the purposes of our class, it's more of a pump, a double pump, right? It's pumping two pumps in parallel. So let's talk about this cardiac cycle, kind of the steps of the heartbeat, right? So during diastole, it's just refilling. This is the longest part. If you were taking notes when you were listening to the lipped up, right? The lipped up comes pretty quick. And then there's kind of a longer pause. That's that diastole when you're refilling. And then there's the next beat, looped up, and then more of a pause. So this refilling is kind of most of the actual cycle. During this time, blood is returning, or I should make this would be blue. Blood's returning from the systemic circuit to get pumped out to the pulmonary side. Blood's returning from the pulmonary side, getting ready to be pumped out to the systemic side. And most of the blood just flows through the atria into the ventricles. So like when the heartbeat actually starts, it's like 85% of the ventricle is already filled with the blood that it's going to be pushing out. So during diastole, it's refilling. Then we're going to have like the SA node fire off. What's going to happen when the SA node fires off?
The atria will contract.
We're going to have atrial systole. Right, and this is going to basically, empty remaining blood into ventricles. Right, so there's some blood that's still in there. So as the atria contract, they'll squeeze their blood into the ventricles. How do we make sure that the ventricles aren't pushing back when they're trying to transfer the blood?
The semilunar valves.
AV valve.
No, this has to go through the AV valve. But what's ensuring that there's no pressure pushing back?
The bicuspid and tricuspid valves.
No, those are the AV valves. Think about our EKG lab on Tuesday.
The delay.
At delay, exactly. Right? The ventricles still have not been excited yet because of that 0.1 to 0.2 second delay of that internodal pathway. So the ventricles are still relaxed. So the atrial systole actually has the ability to transfer blood. We push the blood into the ventricles because the ventricles are still relaxed. They're not pushing yet to push back against it. Right, then there's that, 0.1 to 0.2 second delay due to that little pathway. Then the AV node fires off. And this is going to trigger ventricular systole. When the ventricles start squeezing, there's actually two steps here. And this is part of the reason why I was talking about the blood pressure ahead of this, because if you kind of remember, we're pushing out into the aorta here, or So the aorta. So when the ventricles start squeezing, does the blood immediately start leaving the heart and going into the aorta?
No,
No.
Why is that?
because it has to travel through the lungs first. Or it has to pick up oxygen.
No, it's already picked up oxygen.
Is it because the other valve isn't open yet because it hasn't reached enough pressure?
Yeah, so how much pressure is it gonna need? So which valve and how much pressure do you need to actually open that valve?
Semilunar valves.
Yeah, so to leave the heart, you have to get past the semilunar valve. What's pushing backwards against the other side? Remember, this is like a swinging door. What's pushing on the other side of this door when the heartbeat is starting?
The systemic pressure in the aorta.
And which part of it? We talked about how blood pressure is got two different like values.
Is it the expanding of the elastic arteries?
Let's go back to see what Sienna was saying here. What is the blood pressure as the heartbeat is just starting?
Diastolic.
It's diastolic, right? That was the whole point where I was kind of doing all this. Heart beats, goes up. Then during diastole, when the heart is relaxed and refilling, we get down to diastolic. As the next heartbeat starts, we have diastolic sitting in the system there on the other side, in the aorta on the other side of that semilunar valve. Right? So again, your little valve is like this little swinging door. It can swing open that way, but not the other way. If I have the diastolic pressure on that side, and this is my heart trying to push blood out through this swinging door, how much pressure do I need to actually push out into the aorta here?
Like somewhere over 80.
Yeah, I have to exceed the diastolic pressure, or else the pressure on this side is going to keep the door shut and keep the blood from moving out. Right, so the first part of ventricular systole here is called isovolumetric contraction. Isovolumetric means the same volume. The ventricles are squeezing, they're building up pressure, but there's no size difference of the ventricle as we're increasing the pressure because no blood is actually leaving the ventricle yet. You know, when pressure exceeds the diastolic, then we get ventricular ejection. At that point, the blood can go off into the body because these little swinging doors, now you have enough force on the swinging door to actually push it open and get the blood out into the aorta. So does that make sense?
You know what I have trouble sometimes remembering is that there's blood on the other side. Outside that swinging door, it's there. It's not like it's, there's like blood and then space and like these giant emboli or something. It's just blood throughout the entire system. So it's like when you're saying that it's diastolic pressure on the other side, sometimes I have trouble remembering that just means more blood waiting there.
Right, so exactly.
Pushed out of the way.
Basically, there's blood in the heart, but there's blood in all your vessels as well. So that blood, which has blood pressure as well, which is being maintained by that compression of those large elastic arteries, is there and pushing back with the diastolic pressure. So you need to push back greater than the diastolic pressure to actually get out into there. So does that make sense? I should mention, we talked about what happens when the intrinsic conduction system of the heart is working well. One thing I didn't talk about so much with cardiac muscle is, I need to talk a little bit more about cardiac muscle. Otherwise, this next piece won't make sense. Cardiac muscle is autorhythmic. If you look at cardiac muscle and you look at its membrane potential, there is these leak channels where it just, like, let's say this is threshold. It just automatically starts drifting up. And when it hits threshold, there's an action potential across the sarcolemma and it contracts and it automatically just drifts up. We call this like the pacemaker potential. So if you take a cardiac muscle cell and just leave it there on a dish, it will still be contracting at some regular rate, even without any kind of help. The native rhythm or frequency of most of the muscle cells is pretty low, like 10 or 15 beats per minute, which is not what your heart rate is. That's why the pacemaker node, the SA node, basically entrains everything else. But if that kind of entraining, if that coordination of all of the contraction of your heart muscle gets messed up due to whatever, the heart muscle will still kind of do these kind of beats and quivering. So if you get this kind of uncoordinated quivering of your heart muscle, that's bad. Uncoordinated. What do we call that? If the, rather than getting all coordinated by the, SA node to be atrial systole and then ventricular systole and, your classic thing that we just saw in our lab. What if it's just going?
Is that a heart murmur?
Heart murmur is when there's a leak in a valve. So there's a little bit of backflow and you can hear a kind of gurgling as blood splooshes through a valve when it shouldn't be.
Is it AFib?
Or fibrillation in general. Right, and there's two kinds. You can have atrial fibrillation or ventricular fibrillation. From our discussion just now of the cardiac cycle, which one is going to be more worrisome?
Ventricular.
Yeah, absolutely. The ventricular fibrillation is going to be catastrophic, right? The ventricle squeezing is what's generating most of your blood flow. Right, atria is that extra 15% to push into the ventricles. If your atria aren't really doing their job, your heart's still basically working fine, just not quite as efficient. Ventricular fibrillation is catastrophic because blood is just not getting pushed out into your system. There's no systolic blood pressure. What do they do when someone's in ventricular fibrillation? Yeah, those paddles, the AED, right? It's basically, it's like, your computer freezes and I don't know what to do here. Let's just turn it off and turn it on and let it reboot and hopefully it's working right. So basically that's what those paddles are, is basically like turn it off and turn it on, and hopefully it reboots correctly and gets back into the proper rhythm.
Sorry, just to clarify, that's when the SA node is not functioning correctly?
It's just overall, there's got, you know, the, I'm not sure all of the possible things that can go wrong. I can just say that intrinsic conduction system is not functioning properly for whatever reason.
My dad actually has a permanent defibrillator implanted in his chest. And so occasionally when his heart rate goes whacked, it goes off and it's like a punch in his chest. It's kind of interesting, but.
Right. I mean, if everybody's seen those, you know, kind of action movies where, yeah, it's going to make your, yeah, those are the fancier. So there's pacemakers that are just like replacing the SA node and just keeping a steady beat. But if they're worried about, you know, fibrillation more, they're going to have these fancier ones that actually have these automatic defibrillators that if they detect the rhythms going way off, they can do that little shock. If you have, yeah. Yeah. There were some fun comics about it. All right, atrial fibrillation. We can talk about atrial fibrillation just for a moment because it's actually quite common, AFib. Are you going to be in immediate danger if you have AFib? Are you about to drop dead?
No.
No, you're not. It'll feel weird. The big danger with AFib is that you don't have the irregular flow, you know, from the atria to the ventricles, from the atria to the ventricles. So the blood tends to more kind of pool and get stuck there, kind of little eddy currents or whatever. And therefore is more likely to create a clot, a little thrombus, which then can break free and, you know, become a stroke or a heart attack if it flows off and blocks some vessel. So people with AFib are typically on, you know, blood thinners to reduce the probability of getting an aberrant clot. You know, in addition, there's different ways to try to, what do they call it? Not converge. Is it? There's some, I forget the term for trying to reset the atrial rhythm. Convert. They try to convert. You can convert either there's some drugs that can help bring the rhythms back online, or else there's electrical procedures to bring the rhythms back online. So they do try to bring the rhythms, you know, regularize the atrial rhythm, because one is your heart will be more efficient, and also you'll less of a chance of a blood clot, which can be catastrophic. So fibrillation. So that's this kind of cardiac cycle. And again, with every heartbeat, you're getting a certain amount of blood getting pushed out to the body. And the amount of blood that's getting pushed out into either the pulmonary circuit or the systemic circuit, they call the stroke volume. So, stroke. This is a volume. If I just type it out, because it will be way faster. So it's the volume that gets pumped out of either the left or right ventricle on a single beat. So if I wanted to get the cardiac output, remember this was liters per minute that's coming out. This is our main measure of flow in the overall system. How are we going to get that from this if we have the stroke volume?
Is it stroke volume times resistance?
No, why would resistance be involved? Okay. So if we have stroke volume, which is liters per beat, but we want liters per minute, what do we need?
How many beats per minute?
Exactly. It's just going to be times the heart rate. So this is actually a really important equation. Cardiac output is stroke volume times heart rate. Because cardiac output is really important, kind of coming back, mean arterial pressure is cardiac output times resistance. So when we're talking about things that control our blood pressure, a big part of it is cardiac output. So to control cardiac output, we need to think about stroke volume and heart rate. So all of these equations are going to be at the core. It's pretty simple once you put it all together. Right now, it might be looking kind of confusing or overwhelming. It's like, oh, there's all these equations. And I know people have math anxiety. This is a simple arithmetic that all kind of fits together pretty nicely. And heart rate, right? If heart rate goes up, cardiac output goes up, blood pressure goes up, all else being equal, right? This is going to be one of the ways to increase your blood pressure is to increase your heart rate, because that increases cardiac output, which increases. Another way to control mean arterial pressure is going to be by adjusting stroke volume. So we're actually going to look at stroke volume in a little more detail right now, because these are more things your body can adjust to play around with your cardiovascular physiology. So again, stroke volume, which is the liters per beat. It's going to be end diastolic volume minus end systolic volume. So end diastolic volume is at the end of diastole, at the end of refilling, right before the heartbeat starts. What's the volume of blood in the ventricle? The end systolic volume is how much is left over when we're done with systole. When the ventricles squeeze, they don't squeeze out every last drop. There is some residual blood left in the ventricle after you've squeezed. If you squeeze harder, there'll be less left over. If you haven't squeezed quite as hard, there'll be more left over. So the stroke volume, the actual amount of blood that left the ventricle in that beat, is going to be how much was there when the beat started and how much is left when the beat's done. Does that make sense? Typical numbers here. Oops, millivolts, milliliters. So typically, when the heartbeat starts, there's about 135 mils in the ventricle. By the time the heart is done squeezing, getting ready for the next cycle, there's maybe 65 mils left over. which means how much got ejected? So Aaron's on top of the numbers today. Thank you, Aaron, for responding. About 75 mils per beat. So that is a typical stroke volume. So if we then extend that to figure out, well, what's a typical cardiac output? And cardiac output is just stroke volume times heart rate. So cardiac output, which is again our liters per minute, going to be like 70 mils per beat. A typical heart rate, for you young bucks who are probably in better shape than the average American, probably a little less, but we usually say around 70 beats per minute is a typical resting heart rate. So let's do a typical resting heart rate, which would be 70 beats per minute. Right, and if you multiply those out, typical resting cardiac output is about, let's just say it's around 5 liters per minute. Right, so every minute, but either your left or right ventricle is pushing about 5 liters of blood out into either the pulmonary or systemic circuits. So that's quite a bit when you think about it. And it gets even more intense because we have what's called cardiac reserve. So, would it be possible to increase the cardiac output? Of course, right? How could you increase cardiac output?
Increase beats per minute.
Yeah, we could increase beats per minute. That's going to take it up. What else could we do?
Increase the stroke volume by pumping harder.
So exactly. So if we pumped harder with more, we pushed more, we'd have less left over after the beat. So we could take end systolic volume and bring it down by increasing the contractile strength of the cardiac muscle, which you can do. Like adrenaline will actually cause the same excitation of the cardiac muscle to make it squeeze even harder. So if the heart muscle is squeezing harder, you're going to have less left over after the beat, which means you've increased stroke volume. You can also increase end diastolic volume in a way I'll talk about in a few moments. So anything that increases overall stroke volume or increases heart rate is going to increase cardiac output. You can have this up. So typically, can increase 4 to 5 times. So just you and I, if we're running around and really need to increase the flow in our blood because we're opening up all these capillaries in our muscle beds and stuff, we can have this cardiac output up to 25 liters per minute. Really conditioned athletes, up to like 7 times. It's like 35 liters of blood coming out of either ventricle every minute, getting pushed out into their tissues. Does that make sense? So increasing cardiac output by increasing heart rate, that should be pretty intuitive to you. It's just going to be autonomic control of the SA node, right? Sympathetic will speed it up, parasympathetic will slow it down. I should mention the heart rate, the SA node without any input is around 100 beats per minute. Like if you cut the vagus nerve, the heart will actually speed up to about 100 beats per minute. That's the kind of intrinsic speed of the SA node. The fact that most people's resting heart rate is about 70 beats per minute is because there is typically some parasympathetic innervation to kind of keep it lower. Like if you cut the vagus nerve, we talked about the vagus nerve being the primary carrier of parasympathetic motor. If you cut the vagus nerve, somebody's heart will actually speed up more to this because that's what the SA node is going to do without any kind of modulation. But obviously we can change beats per minute just by sympathetic and parasympathetic. Changing stroke volume is a little more involved. Let me talk about that right now. So one way to change stroke volume is by, if we want to increase stroke volume, we could increase end systolic volume. This can happen by increasing how strong the cardiac muscle squeezes. For instance, so like epinephrine. Right, so then that would actually take this down. Like, so if in the presence of epinephrine, the cardiac muscle will actually be squeezing harder than it would have otherwise, and therefore we'll have less left over at the end of the beat, which means we've increased the stroke volume, right? We pushed more blood out into the system. Changing end diastolic volume is a little trickier to think about. So you have to remember that the blood is in a closed circulation system. So I'm going to kind of simplify this because it's going to be easier. Let's just do one circuit instead of two circuits. Oh my God, I can't spell system. So the blood is all in here. Typically, we're going to talk about the veins have very low pressure because there's no, the main pressure is being generated on the arterial side. There are things that keep the blood moving back to the heart, but typically there is a lot more of your overall blood volume sitting on the venous side versus the arterial side. So at any, we keep pushing blood out into here and it keeps coming back on the venous side, but typically there's more blood on this side slowly making its way back to the heart. Veins, though, they're vessels, they have muscle, smooth muscle in their walls. If we want, we can actually increase the venous return by contracting the walls of the veins and kind of changing how much blood is actually allowed to kind of stay circulating in that venous area. So you can increase venous return. Meaning we're pushing the blood out and more of that blood is actually making it back to the heart because we've actually kind of constricted the veins, the walls of the veins, so there's a little less room for the blood to just kind of hang out. Right, so that's something your body can do, is kind of constrict the veins, which is going to increase the venous return, how much blood is actually making it back to the heart from the veins. You know, this is going to increase our end diastolic volume, which ultimately is going to increase stroke volume, which is ultimately going to increase cardiac output. I should do this way. So increasing venous return is going to increase the end diastolic volume, which will then increase the stroke volume, which then can increase cardiac output. So you can see how all these things get interrelated. Like this weekend, I highly recommend you kind of just write this stuff out and see how they all connect. You know, one of the terms they talk about is concept mapping, kind of just putting it all out. You know, if I was going to do a concept map, you know, mean arterial pressure is cardiac output times resistance. The cardiac output is stroke volume times heart rate, but you know, stroke volume is end diastolic volume minus end systolic, you know, end systolic volume. You know, so you can see how any one of these things can ultimately be manipulated to ultimately affect the mean arterial blood pressure, which is going to be something we're going to be having to track in this class, all the different ways that we're controlling blood pressure. The one other thing that is Sienna?
One thing I don't understand is you're saying you can increase the venous flow back to the heart by constricting, but doesn't the constriction of the vessels create more resistance?
In this case, it's more just about having, there's so, yeah, first veins are much bigger and to start with. We'll talk about veins. So this is one of these cases when you're talking about like the main arteries leaving the heart and we talk about changing the diameter, you definitely think about it mainly being about resistance. In this context, it actually is not so much about the resistance. It's more just about having a little less room for the blood to be pooling around, so it ends up finding its way back to the heart.
It's kind of like the constriction of the aorta.
No, no,
No.
the aorta doesn't constrict. The
Well,
aorta,
after it balloons out,
that's more compression.
okay.
This is actually muscle. But it's the context. It's like right. If you hear your cat meowing as you're getting the food bag out, you kind of know the cat's just excited because it's gonna get breakfast. But if it's in the middle of the night and the cat is meowing, you know, in that different context, it's probably a warning that there's something wrong, right? So the same thing can have different implications in different contexts. So when you're looking at changing of the diameter, and we're looking at it in terms of major arteries leaving the heart, like the carotid and the blah, 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 it's going to be a very different implication then. But I can see why you're thinking that.
I get it intuitively how constricting the veins would increase flow though.
And that also, we can talk about the Frank Starling law of the heart. and this is two people. There was a dude, Dr. Frank and Dr. Starling. It always sounds like it's a guy named Frank Starling. This is basically a observation that the heart will pump out any amount of blood within reason that's returned to it. So if you increase venous return, the blood is gonna, that increased amount of blood returning to the ventricles will still get pushed out into the body. It's actually worth talking a little bit more about cardiac muscle. So I said cardiac muscle is autorhythmic. It can also have varied contractile strength. So I mentioned already, for instance, the presence of epinephrine will, through mechanisms we'll talk about later, increase the strength that the cardiac muscle is pulling when you excite it. Another thing that can increase the contractile strength is pre-stretching. So if the muscle is stretched out more when you start the contraction, it will actually squeeze harder than it would have if it wasn't pre-stretched. That is going to be responsible for this Frank Starling law of the heart, right? I have my ventricle here and here, and I've increased my venous return, so there's even more blood in here than normal, which means these are even more stretched out than normal, which means when the heartbeat starts, the cardiac muscle will squeeze harder than it would have normally, so we'll still be squeezing out that extra volume of blood that's sitting in the ventricle. Right, so if you fill the thing up more and stretch it out more to start with, it'll squeeze even harder to make sure that blood leaves. So that's the Frank Starling law of the heart. That makes sense. The other thing we should talk about while I have these pictures up. So this idea of the closed circulatory system is got one other super important implication. Let's get rid of this other stuff so we can stay on what's important. So because it's a closed system, the other thing that's really going to be important in determining the overall pressure inside all of these vessels is just how much fluid is inside compared to the overall total room inside, right? If I increase my blood volume too much, there's just going to be too much pressure because it's just going to be pushing against all the walls. If I have too little blood, then everything's going to be floppy because it's not going to be like, pushing enough. So blood volume is going to be the main long-term control of overall blood pressure. So this is really important. When we get into control of blood pressure, which we're going to be getting into now, there are all sorts of short-term controls of blood pressure. In the short term, if I wanted to control blood pressure, I could change this, or change this, or change this, or change resistance by constricting vessels or relaxing vessels. There's lots of ways to do this short-term control of vessel in the moment. But for the long-term just control of your baseline blood pressure, it's actually blood volume. So that's really important. When we look at long-term control of blood pressure, just the way your body does it, it has a lot to do with your urinary system, controlling your kidneys, either conserving water or peeing out more water to adjust your blood volume. And that is an important part of your blood pressure control, is actually talking to your kidneys to conserve water or to let go of water, which is going to control blood volume. You know, if people have high blood pressure, one of the what kind of drugs do people take for high blood pressure?
Beta blockers.
So one thing they do is take beta blockers. Beta blockers, which block like epinephrine. Epinephrine is going to keep the heart rate down, because remember, epinephrine speeds up your heart rate. So beta blockers are beta adrenergic antagonists, right? They're blocking the beta adrenergic receptors. They're going to keep your heart rate down. We also talked about how adrenaline or epinephrine increases the contractile strength of the heart muscle, decreasing the end systolic volume. So it's actually going to keep the end systolic volume higher, which is also going to decrease the stroke volume. So the beta blockers decrease the heart rate, decrease the stroke volume, which is ultimately decreasing the cardiac output, which is going to keep your mean arterial pressure down. So you can see why all these equations are actually important to understand. This is the reason beta blockers work, is pretty straightforward once you kind of get all these relationships in your head. That's why I was saying, like, over the weekend, writing out some concept maps of how this all relates and making sure it kind of makes sense is going to be a useful thing probably. So beta blockers. What's another important drug people take for high blood pressure?
Diuretics.
Diuretics. Those are going to work by reducing blood volume, which again, we said is the main long-term blood pressure control. The diuretics are going to make you pee more. If you pee more, then you've got less overall volume inside this whole system here. If you have less overall volume pushing out against the walls, you're going to have less overall resting blood pressure. So diuretics are another important way of controlling high blood pressure hypertension. Now, there's one other class of drugs, the ACE inhibitors, that we're going to talk about as well. But we need to talk about the renin-angiotensin mechanism before we can get to those. But again, just reiterating, because I know this is not always intuitive for people, that the blood volume is so important for your blood pressure, and that your kidneys and your urinary system are so critical in terms of the control of your blood pressure. So, all right. Oops, that's not where I meant to go. So this control of blood pressure is obviously critical for, you know, your body to make sure the blood's getting everywhere it needs to go. So we're gonna kind of talk about, you know, some of the ways that your body does control blood pressure. And here I've already talked about a bunch of them, but I'm going to talk about a little more formally some of the basic mechanisms for adjusting your mean arterial pressure. Again, things like heart rate, resistance. Again, by how would we if your blood pressure was dropping, how would you try to bring it back up? This happens when if you've been lying down, there's not much blood pressure you need to get the blood into your brain because the blood's not fighting gravity. And then you sit up, all of a sudden you've got this huge drop in blood pressure going up into your brain, and you need to get it up or else you are going to experience syncope, fainting. So what is your body going to do like instantly to try to make sure you don't have a head rush and blackout and face plant as you get up from bed? And let's start right here.
Increase your heart rate.
So one thing you can do is increase the heart rate, which is going to increase the cardiac output, which is going to bring up the blood pressure. So actually within a beat even of getting up, your heart rate speeds up for a moment to help keep the blood pressure up. What else could you do in that moment to keep the blood pressure from dropping? And look at this equation.
Reduce resistance by dilating your blood vessels.
Would that keep the blood pressure up? What would reducing resistance do to your blood pressure?
Oh, increase resistance.
Yeah, you actually constrict like your carotid arteries, increase resistance. So if you try to increase the flow and increase the resistance in that second as you get up, and that's going to keep the blood pressure up as all of a sudden the blood has to fight gravity. As you get older, these little reflexes are slower and you're more likely to experience a head rush or pass out actually as you go. The idea they call orthostatic hypotension. Orthostatic means straight, standing up basically, hypotension like low blood pressure. As you go from lying to standing, you experience a drop in blood pressure going up into your brain, and you need to make a quick adjustment so you don't black out. That is what we call like a neural mechanism of blood pressure control. So blood pressure control, I'm going to clear all these drawings here. When we talk about blood pressure control, we have neural mechanisms. And we have chemical mechanisms. We also have short-term versus long-term. And these all often overlap. Right, you can use the nervous system to trigger the release of renin, which is a chemical mechanism. Renin-angiotensin mechanism has lots of short-term things, but also triggers lots of long-term things. So we're going to talk about a few different examples of these systems to regulate your blood pressure. But we'll see that some of them are like this vasomotor reflex or baroreceptor reflex, which is basically kind of what I just mentioned with orthostatic hypotension, is kind of a neural short-term mechanism. But then some of the other important ones are weird mixes of everything. So let's talk about the baroreceptor reflex. Let me look at the time. Yeah, I'll give you one example here and then we'll take a break. So an example of a short-term neural mechanism. The baroreceptor reflex. So baroreceptor, that means a pressure sensor. What do you think a baroreceptor actually is? How is your body going to get information about your blood pressure?
By lying there in an arterial wall and feeling how hard the blood is pushing back on it.
Yeah, and it's actually a stretch receptor. Is how stretched out. Like the higher the pressure, the more the artery gets stretched. So these are basically stretch receptors in the arterial walls. And typically in the major ones, like in the carotid arteries, the aorta. You know, carotid artery is the main artery going up into your head. The aorta is the main artery feeding most of your body. So you've got these stretch receptors in the walls of these main arteries that are giving feedback, like, is there adequate, what's the current pressure right now? And the baroreceptor reflex, you know, if blood pressure is going down, what is something your nervous system can do kind of instantly to bring the blood pressure back up?
Constrict that artery.
Exactly. This leads to vasoconstriction. Let me write this better. So vasoconstriction of like kind of the major arteries leaving the heart there. That's, you know, assuming the flow is staying relatively constant, that's going to bring the blood pressure up. Right, this is that sympathetic mainly. Right, we talked about vasomotor. Motor just means muscles. Vasomotor is just the smooth muscle in the walls of the blood vessels. You constrict the vessel and it will increase resistance, which increases blood pressure, right? Mean arterial pressure is cardiac output times resistance. So if resistance goes up, blood pressure goes up. So this is, you know, one example of a short-term neural mechanism to adjust your blood pressure, is just vasoconstriction to bring the blood pressure back up. As I was saying, for that orthostatic hypotension, when you're standing up after lying down, this is happening, as well as have an instant speed up of your heart as well, because that is also going to, you know, increase cardiac output, which will also help keep your blood pressure up. So baroreceptor reflex is one example here. The other, I'm looking at the time. Yeah, let's take a break so you guys don't get glazed over. And when we come back, I'm going to spend a little bit more time just doing one last of the major regulators of blood pressure, the renin-angiotensin mechanism. Before we leave the heart, we've been talking about heart rate. I realized one thing that's actually be interesting just to mention is, you know, heart rate we said is typically around 70 beats per minute. But it's not, like they say, the heart is not a metronome. If you look at the heart rate, it's about 70 beats per minute. It actually varies with breathing, but it also varies because there's this constant push and pull by the sympathetic and parasympathetic. So it's speeding up, slowing down. So people talk about heart rate variability. So if I did a little chart, like the percent, let's say 70, 60, 80, if I had my heart rate, quote unquote, 70, it would be like, it's mainly around 70, but sometimes it's a little faster, sometimes it's a little slower. So it's constantly kind of speeding up and slowing down a little bit. So it's really kind of smeared out around 70. This is something that people measure. It's one of the dependent variables in the experiments that I've been doing with the MDMA patients, is looking at their heart rate variability before and after they go through the treatment. Because heart rate variability is actually correlated with like anxiety and things like that. In general, people who are have more anxiety disorders have less heart rate variability. Things are kind of locked in more. People who are healthier, more well adapted and adjusted tend to have greater heart rate variability. There's more kind of wiggle, probably because your nervous system is more flexible and constantly a little more, a little less, a little more, a little less, versus like, just hold on. So that idea of your having a really steady heartbeat is actually a bad sign. You want your heartbeat to be, in the zone, but then wiggle around it. So it's kind of an interesting thing. All right, but what I was saying, we're gonna finish today by just adding in one more super important thing that controls your blood pressure, called the renin-angiotensin mechanism. So let's do that, and then we'll get into the lab. Sometimes they call it the RAA, renin-angiotensin-aldosterone mechanism. You'll see why that would be appropriate as well. So basically, renin, this is released by the kidneys. We'll meet the kidneys in more detail in a couple of weeks. We'll meet the you've probably met the nephron in your other classes. The juxtaglomerular apparatus of the nephron is what is releasing renin. Don't worry about what any of that means if it doesn't mean anything yet. Let's right now just know it's in the kidney. And it's typically released when your blood pressure is dropping. So it's going to do a bunch of different things that help try to bring your blood pressure back up. It can also be triggered by just the nervous system. Like your sympathetic nervous system can actually trigger the release of renin directly if your brain wants to pump up your blood pressure for whatever reason. So it can also be triggered by the nervous system, but more typically it's released in response to drop in blood pressure. So renin is going to just start a whole cascade of events. In and of itself, it isn't doing anything directly. What it's doing is catalyzing and beginning the whole chain reaction. So in your blood, you have all sorts of plasma proteins, right? You know, in your blood, you have the blood cells, but also dissolved in your plasma are all sorts of different proteins responsible for all sorts of things. We're going to be meeting lots of them as we go through the semester. There's proteins that are involved in binding, you know, nonpolar things to transport them in your blood. There's going to be proteins in your blood that are there that are part of your immune system. There are going to be proteins in your blood that are going to be important for blood clotting. There are proteins in your blood that are part of the renin-angiotensin mechanism, in particular, angiotensinogen. So angiotensinogen is just a protein in your plasma. Normally, so anything with the gen at the suffix means it's like something that is going to create something later. It's in an inactive form. Angiotensinogen is just floating around, waiting for its moment. Renin is going to catalyze the conversion of angiotensinogen into angiotensin 1. So angiotensin 1 is now in there. It's still not the active thing. Angiotensin 1 is then going to get converted into another protein called angiotensin 2. And that is catalyzed by by an enzyme that is anchored particularly in like the lung tissue. It's something called angiotensin converting enzyme. So called ACE. So renin is catalyzing the conversion of angiotensinogen into angiotensin 1. And then this ACE enzyme is converting angiotensin 1 into angiotensin 2. Just as an aside, COVID, the coronavirus that causes COVID is binding the ACE on the cell membranes. There's different versions of the ACE membrane proteins, ACE receptors, and there's one of them that is the way that the coronavirus actually gets into your cells. So you might have been reading about ACE in that context more recently, but here we're going to talk about what ACE is doing when it's doing its normal thing rather than providing a backdoor for you to get COVID. And again, it tends to be located in the lungs. So as the blood is circulating through your body and goes through the lungs, the angiotensin 1 gets turned into angiotensin 2. The angiotensin 2 is going to be the thing that makes stuff happen. And again, it's going to do things that are going to help bring your blood pressure back up. And it's going to be a bunch of things, both long and short-term controls that we've been talking about. So let's talk about angiotensin 2. In and of itself, it's a potent vasoconstrictor. Right, so if that's all it did, that would help bring your blood pressure up. Vasoconstriction, increased resistance, increased blood pressure. But it does a lot more. So one thing, it's a potent vasoconstrictor, triggers the release of a couple of hormones. Antidiuretic hormone from the kidneys. Not from the kidneys, from the pituitary. Talks to the kidneys. And aldosterone from the adrenal cortex. So these are hormones. We introduced hormones when we talked about epinephrine, basically signaling molecules that get dumped into your bloodstream and then will have an effect on any cell with the appropriate receptor. So let's look at how ADH and aldosterone are going to help increase your blood pressure. So one thing is ADH is another potent vasoconstrictor. Constrictor. Again, so in fact, another name for ADH is vasopressin. Vessel pressure. So far we're still in this kind of more short-term control, right? Both angiotensin 2 being a vasoconstrictor and ADH being a vasoconstrictor are going to be increasing the resistance, therefore increasing the blood pressure. I mean, but from its name, what else does ADH do?
Stops you from going.
Stops you from going wee-wee, right? It's gonna be antidiuretic, you don't pee as much. So it talks to the kidneys and tells them to conserve water. Now, why is that going to be helpful to bring your blood pressure up?
Increases
Volume.
Exactly. It's going to keep the blood volume up. And we said that the blood volume is the main long-term control of your blood pressure. It also makes you thirsty. What part of your brain do you think it's talking to make you thirsty?
hypothalamus.
Uh-huh. It triggers the hypothalamus to make you thirsty. Why is being thirsty going to help your blood pressure go back up?
Drink more water, increase volume more.
Exactly. You're going to bring more water into your body and increase your blood volume, which is also going to keep your blood pressure up. So ADH is doing these short-term vasoconstriction as well as long-term blood volume things, both in service of bringing your blood pressure back up. Aldosterone talks to the kidneys, tells the kidneys to conserve salt or sodium. Again, why is that going to be helpful? Keeping the salt in your body?
Because the water is going to be drawn to it.
Yeah, exactly. It's osmosis, right? The more you pull the salt back into the body, the more the water is going to follow and stay in your body, right? So conserving salt, increases the reabsorption of water. Right, so we're going to talk about this when we do the urinary system in more detail, but antidiuretic hormone basically tells the kidneys to hold on to water But antidiuretic hormone in conjunction with aldosterone is even more efficient at pulling water and keeping it in your body. So aldosterone and ADH both work together to maximize the conservation of water and keep it in your body and keep your blood volume up. And ADH also triggers your thirst centers to bring your volume up. So those are like these long-term controls. And then ADH, along with the angiotensin II itself, are potent vasoconstrictors, which are going to be more short-term. Put the resistance up, which is going to put the pressure up. So this brings us to a third class of very commonly prescribed blood pressure medications. We talked about the beta blockers, which are the adrenergic antagonists. We talked about diuretics. What's another major class of blood pressure control meds? Someone probably knows somebody on these things. The ACE inhibitors. Have people ever heard of these? They're very, very commonly prescribed. Most of their names end with ill, like rampilil and this allele and that allele. Why would inhibiting ACE be a good move if you're trying to keep blood pressure down? So
It prevents the angiotensin II production.
exactly. So without ACE, if we're inhibiting ACE, then we never make angiotensin II. And if we don't make angiotensin II, why is that helpful?
You have none of the things that bring blood pressure up.
Yeah, that means we're not going to have angiotensin II, which is a vasoconstrictor, and we're not going to release ADH, which is a vasoconstrictor and also increasing your blood volume, and you're not going to release aldosterone, which is also helping increase your blood volume. So by blocking ACE, you are blocking all of these processes that would serve to increase your blood pressure. So yeah, make sure that makes sense. Like why an ACE inhibitor is an important intervention to help keep your blood pressure down. Because normally your kidneys just naturally would be releasing renin in response to increases of blood pressure. I mean, not decreases of blood pressure, right? So when your blood pressure drops, typically renin is released, and when renin is released, all of this stuff happens. So if we don't have this happening, the blood pressure is going to stay lower.
I have a question.
Sure.
So you have angiotensin 1 as kind of this middleman in this whole process. Does it mostly serve to amplify the effects that start with renin and lead to all the other angiotensin 2 and ADH and aldosterone? Is that mostly the purpose of it? I mean, greater control, I guess, too, but like mostly.
I would say, maybe people who look at this more deeply see more of a role of it. I just see it as through the evolutionary hodgepodge of the development of these systems, it ended up being this multi-step process instead of a lot of things in your body are just kludges, things that are on top of other things. Kind of, if you think about plants, right? If you look at just a daisy, a photo system one, photo system two. Why do you need two photo systems? It's because at some point the original photosynthetic pathway got, kind of new kludge on top that made it all a little more efficient. But a lot of things in your body. Think about cellular respiration. What a mess it is. Yeah, it does the job, but if you were designing it from scratch, that's probably not how you would do it. It's just, so I think.
So it's like the Rube Goldberg machine that you showed us.
Exactly.
Okay.
That's how I view it, at least. I mean, it's obviously my, what do you call it? I can't say for sure, but it's definitely through the lens of evolution, it makes sense that things are going to ultimately end up functional, but not necessarily super pretty or efficient. So,
okay.
right, it's like, why, men have nipples even though they don't have to nurse babies, but it doesn't hurt, so it doesn't have to disappear from the species or something, right? So it's like, there's lots of stuff that ends up kind of left over through different things, just as long as it's
not getting in the way.
As long as it's not getting in the way, then there's no reason to change it.
Huh.
So
interesting. Cool. Thanks.
All right. Are there any questions about this renin-angiotensin mechanism? Because it is important. Okay, let me show a couple of other things here. I'm assuming everybody ended up getting the textbook, because I've had some people send me questions that are like just straight out, like if you just like open the book and it would be just sitting there right in your lap. So I'm not sure everybody actually has a textbook. Hopefully you guys do. But kind of looking in here, it's worth just reviewing, like over the summer, just over the weekend. Remember the main circuits. Remember, some of the circuits are a little more complicated, like these portal systems. Talked about portal system, hepatic portal system. There's a portal system in your kidneys as well, part of the nephron. Remember, we're going to get there later. Don't worry about that. We definitely talked about I didn't give it its name. If you talk to Tina did her, like, some of her graduate work looking at Poiseuille's law. So she says it more than I do. Also, I have trouble pronouncing it. Poiseuille. That's that thing saying that the resistance to flow is inversely proportional to the fourth power of the radius of the tube. So, but all this is just kind of showing, let's see. What else do I want to have in here? Well, this is kind of showing the development. Again, it's just kind of wild because it has to do its job when you are a little embryo less than a month old, and it never stops until you're dead, but it obviously changes form pretty dramatically without ever stopping doing its job. So it's like, I just find it super impressive. This is more of a realistic picture of a heart where the aorta is coming out the top. Here you can also see that fibrous skeleton of the heart that electrically isolates the atria from the ventricles is also reinforcing all of the valves which are sitting at that same plane. Semilunar valves, bicuspid, tricuspid valve. But again, for this class, you don't need to know the anatomy any more than kind of those silly cartoons I've been drawing on the board. Yeah, we're going to talk a little more about cardiac muscle cells later. This is where I was talking about that pacemaker potential. Even if you don't do anything, the voltage on the membrane just moves up until it hits threshold. So this is, I didn't talk about it, but I'll just give you a little heads up. The dicrotic notch. So this is this increase in your blood pressure as the ventricles squeeze. And then as the ventricles relax, the pressure starts dropping. And at some point when the pressure drops low enough, the semilunar valves will slam shut. In that moment when the semilunar valve slams shut, there's this little back pressure that happens, and the pressure actually bumps up a little and then continues dropping down towards the diastolic pressure until we get to the next heartbeat. So dicrotic notch happens because as the semilunar valve slams shut, there's a little hiccup of back pressure that ends up, like think about, right, if you're kind of blood's moving out the aorta, and then there's that little closing all of a sudden, and there's like this little back pressure thing. Not worry about that. Well, here I was saying things that influence stroke volume. So this is stuff you should think about. You know, Frank Starling law of the heart. Stroke volume is proportional to end diastolic volume. The heart pumps out all the blood returned to it. So if part of stroke volume, if you return more blood to the heart, the heart's going to squeeze harder and squeeze more blood out. You know, stroke volume is affected by venous return. You know, end diastolic volume is determined by venous return, which is affected by sympathetic innervation of the veins. We haven't talked about this other stuff yet. You know, length tension relationships determined by volume of blood at the beginning of contraction, degree of stretch is called preload. And that was, I was saying, the more the heart muscle is stretched out before you start the beat, the harder it's going to squeeze and more blood's going to leave. So when you look at this these are all charts from your book. So you can look at this stuff and then try to connect it and see if it all makes sense. This is showing with norepinephrine. There's more stroke volume because with norepinephrine, you've got more squeezing, more contractile strength. You know, don't get too caught up in here. Don't worry about comparing cardiac muscle and skeletal muscle and things like that. We haven't even talked about skeletal muscle yet. You know, catecholamines. Catecholamines I talked about are basically adrenaline and noradrenaline, epinephrine, norepinephrine. You know, they bind these beta adrenergic receptors and ultimately create these more forceful contractions. Again, don't worry about the detail. This gets into stuff you don't need to know. Cardiac output. This should look familiar. Heart rate and stroke volume. Heart rate is going to either decrease due to parasympathetic innervation or increase due to sympathetic innervation and epinephrine, adrenaline. So those are things that can control heart rate. Stroke volume is controlled by force of contraction in the myocardium, influenced by the contractility. We said that sympathetic can increase how contractile it is, how hard it squeezes by decreasing end systolic volume. We can increase it with venous constriction, which increases increased venous return, which means we've got higher end diastolic volume, which is going to help increase stroke volume. again, we don't worry about this other stuff, but you should look at these charts, and at least overall, this should make sense if you look at this. Like, I definitely know for myself, when I first started learning this, I would just look at these charts and keep looking at them until they started making more intuitive sense. Because I know in the beginning, it's just a whole lot of things. That's why I was saying doing these concept maps and writing things out, how they all relate to each other. Looking at these relationship charts is useful. So that's the one for cardiovascular. The other one is just all about blood pressure. This is also from your book. Kind of giving you the sense of it's this closed circulatory system. You know, systemic veins serve as an expandable volume reservoir. So if you constrict these veins a little bit, there's less of a reservoir there. So more of the blood is returning to the heart, that increased venous return. Here you can kind of see, like, it's more just about how much room is there. It's not about changing the pressure at that point. Here it says the big elastic arteries are the pressure reservoir that maintains blood flow during ventricular relaxation, right? We talked about having the big elastic aorta and pulmonary arteries. By having them stretch out during systole, then they can compress and keep the blood pressure from dropping to zero during diastole. Again, the water weenie effect is what I think of it as. We'll talk about blood vessels later. We'll talk about that stuff later. We'll talk about, yeah, we'll do this next week. So factors that affect blood pressure. Mean arterial pressure. This is the kind of graph. If you see this, you should make sure it all makes sense. Mean arterial pressure determined by blood volume. Blood volume, which is going to be a mix of fluid intake and fluid loss, right? Fluid intake that we talked about, your ADH in response to the renin-angiotensin mechanism making you thirsty to help bring your blood pressure up. Determined by fluid loss. Your kidneys are going to be an important player of mean arterial blood pressure by letting go of more or less water and adjusting your blood volume. You know, mean arterial pressure determined by your cardiac output, right? We talked about it's cardiac output times resistance, basically. And cardiac output is heart rate and stroke volume. And we just talked about all these things that affect stroke volume. You know, it's determined by resistance to blood flow, which is mainly determined by the diameter of the arterioles, like in the more short term. You know, we said there are other things like, you know, viscosity and things, but, you know, in terms of blood pressure control, we're usually talking about the diameter of the vessels, vasoconstriction, vasodilation. You know, it's also determined back relative distribution of blood between arteries and veins. This is kind of related to, you know, venous return, which is more about stroke, you know, cardiac output and stroke volume. But so you should look at this chart, like figure 18b, and you should be able to look at each of this and like, oh yeah, no, that makes sense. I kind of see that. Not I kind of, like by the next exam, it's like, I totally see that. So just kind of putting that out there. There's a lot of moving parts, but they all lock in together, and it makes sense once you start putting it all together and seeing how they all interrelate. You know, compensate, blah, blah, blah. And so this is, we talked about short-term and long-term. You can have short-term things where you just do vasodilation or speed up or slow down the heart For the long term, it's really going to be more about the kidneys changing blood volume. But you can change blood pressure pretty quickly in the moment, but not necessarily in a sustainable way. I can give you an example. There was one time when I was changing the compost, and it turns out that all the food in the compost had been molding like crazy. And there was a huge, it was basically like all of a sudden there was a cloud. And I just inhaled a huge amount of mold spores. And which in the big picture, mold spores aren't really that bad for you, except I had a major allergic reaction to them. And part of that allergic reaction was a major vasodilation. Part of like inflammatory response is vasodilation. So all of a sudden, I had a lot of vasodilation. All I knew, not dilation. All I knew is all of a sudden, my heart was beating like crazy. My resting heart rate's usually around 60, and my heart rate was something around like 160 or more. I was like, what the heck? And then I was like, and I was all flushed and red because all of my blood vessels are dilating. So I'm all flushed. And now I'm kind of terrified, like, what's going on? And as I'm realizing major vasodilation, I actually have a little blood pressure thing. My blood pressure was fine. My blood pressure, I'm going to pretend it's probably a little worse than this. Let's say it's that. So I measured my blood pressure and it actually was okay. But my heart rate was like, 170 beats per minute. So what do you think was going on?
Your heart rate had to increase to compensate for your blood vessels dilating as a result of, as a reaction to the mold spores, and your baroreceptors picked that up and kicked the heart into high gear so that you wouldn't pass out.
Yeah, exactly, right? The mean arterial pressures, cardiac output times resistance. When I had this massive vasodilation in my body, the resistance plummeted, which means that blood pressure is going to plummet. So the body is like, what the heck are we going to do? We need to kick up cardiac output. And a big part of cardiac output is heart rate. So one of the major things my body did to keep my blood pressure relatively stable was just, triple my heart rate almost to keep my blood pressure up, which is keeping the cardiac output up, which kept me all right until I got over, a few hours later, everything was resolving. But it was kind of freaky. I was kind of glad I actually knew about this stuff so I could relax once I realized my heart rate or that my blood pressure was actually stable. So, but that's like a short-term kind of thing, right? So your body has ways to adjust your blood pressure by constricting or dilating vessels, speeding up or slowing down your heart. But in the big picture, it's not going to be a sustainable thing. In the big picture, the control of your blood pressure is going to be by maintaining the appropriate volume of your blood via your kidneys. So just kind of putting that out there, that idea, there's these short-term ways to adjust things in the moment. But in the long term, you obviously, I wouldn't want to like have my heart rate at 170 for the rest of my life. That's not a good thing. Anyway. Right, so again, if, let's see, hyperemia, we don't have to talk about that. This is just kind of showing flow. You know, flow is driven by pressure. You know, pressure is basically the mean arterial pressure. How do I look in here? Yeah, this is just another way of looking at the same basic stuff. This one doesn't seem as interesting. The one about blood pressure is very interesting. Like this one, this figure 15.8b, you should definitely understand this one. If you look at this one and there's anything on here that doesn't really, it's like, I kind of see it, but I don't really own it, you should go and own it. So that's what I would say. Anything on this should be something you know well. Some of those other ones get into some stuff that's a little more tangential. We'll talk about this. I will mention there's different amounts of blood that goes to the different tissues. Which are the organs that you think always get their full share of blood? Even if you're having cardiovascular problems, which organs do you think are always going to get the blood they need?
The brain and the heart.
Yeah, the brain. If the heart doesn't have what it needs, nothing's going to get anything, right? The heart's driving the whole thing. And the brain in particular is very intolerant of ischemia. Within minutes, you're going to have neurons in your brain starting to commit suicide. So we'll also talk about so we'll talk about more about orthostatic hypotension. This is just, we just talked about. Yeah, so there's more to talk about that we are going to continue, but we're going to take a break for today. Well, this is kind of also atherosclerotic plaques. Like, why do you having hardening, these atherosclerotic plaques in your arteries is going to impinge the diameter. So it's going to increase resistance. It's going to need more blood pressure to get stuff moving through the vessels, or up to a point. If it's too constricted, then you're not even going to get the blood flowing. They're just blocked off. Like when people get coronary bypass operations, it's because their coronary arteries are so blocked off that you don't get adequate blood flow actually getting into the heart. These little plaques, we'll talk about them more. They also make the arteries less flexible, which obviously is not so good because the pulsing pressure is going up and down and up and down and up and down. And if you've got some flexibility, it's like, all right, I can do this. I can do this all day. But if you are losing that flexibility, then it's like, it's more likely to cause damage because it's like impacting without and having that little bit of give. So having the kind of hardening of the arteries, the less flexible arteries is not so good. So, all right, blah, 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 blah.